Near majestic Mount Rainier in the Pacific Northwest, dedicated scientists from Unigen are discovering the beneficial powers in natural products. Utilizing sophisticated equipment and a pharmaceutical approach, we discover and patent unique active ingredients. These proprietary ingredients are thoroughly tested for safety and efficacy at the molecular, genetic, and human levels. These bioactive compounds demonstrably enhance health and well-being when formulated in functional foods, nutraceuticals, and cosmeceuticals. Come learn more about our unique ingredients, supported by our strong science, to better differentiate your products from your competition.
Hello, my name is Qi Jia, Chief Scientific Officer of Unigen. Today I'd like to present to you the PhytoLogix technology platform and the discovery process to find novel natural ingredients for functional food, cosmetic, and dietary supplement. PhytoLogix is a technology platform which including bioprospecting, library generating, high throughput screening, structure dereplicating, and product developing. Unigen targeted collecting more than 20,000 medicinal plants to make 20 or more 100,000 of extracts and millions of fractions and many thousands of compounds, and to screen the library against the different biological and biochemical targets to develop novel ingredients out of this process. PhytoLogix discovery process is starting with screening the library generated in the platform and screen them over many different type of biochemical, biological targets. In the middle of the discovery process, the natural components can be developed as a standardized extract to be utilized in functional food, dietary supplements, and cosmetic industry. And also, we can further develop the active components as a drug leads and into herbal medicine and pharmaceutical prescription drugs. In past 10 years, Unigen signed more than 10 plant collection agreements around the world, and those botanists went to mountains and oceans and forests to collect documented medicinal plants and bring the material with botanical taxonomies and collection form to Unigen for us to process it. PhytoLogix library has a collections of many thousands of indigenous plants from India, China, and Korea. And also we do have several thousand of plant collection from African countries. And we collected the medicinal plants from Rocky Mountain and the Pacific West regions in the United States. We started the marine collection from Pacific and Atlantic Ocean. And we have a collection several thousand per year ongoing to bring the material to here. Unigen has a high throughput technology for library generation. We are using an accelerated extractor to process 12 medicinal plants or marine every day to generate 24 extracts. Every single material will be extracted with organic solvent followed by aqueous solvent. Every single extract will be processed with a high throughput fractionation system to make 88 fractions. So every single plant we collect will generate 2 extracts and 176 fractions. As of today, we have many thousands of extracts to be generated, and hundreds of thousands of fractions has been prepared. Those samples are stored in 96 well plates, ready for high throughput screening. In terms of the target selection, Unigen look into the state of our health and customer needs worldwide. And one of the examples is look into the pre-diabetic conditions such as overweight, obesity, and those sub-health states, and we dissect those conditions into individual pathways. We develop biological and biochemical target for screening. PhytoLogix discovery process is like a funnel, and after we identify the target, develop the mechanism of action based biochemical or biological screening model. We validate the model, and we loaded our libraries on the top of the screening, and the hit coming out of screening will be confirmed on different systems, and bring those hits into in vivo and in vitro evaluation, and running the safety, efficacy, and bioavailability study. By the end of it, those material will be evaluated on human clinical trials. And most of the screening model in-house or with the collaborators are protein-based receptor binding or enzyme inhibition assays. And we also can develop cell-based assay and using very complicated systems such as beta-lactamase gene reporter assay. We overexpress the receptor on the cell membrane, and also we insert the gene to express the beta-lactamase enzyme, which can turn the substrate signal from one color to another, so we can detect the biological functions of extract by triggering the response from receptor to the signal changes.